get started with this issue in Science a half a year ago, which probably all of you know, since you are devout light experts. And it sort of highlighted the issue of light pollution. It was triggered by this study by Chris Kuba here, who did a citizen science project where thousands of participants from across the world, as you can see here, from different parts of the world, over 12 years, kept continuing to collect data on light pollution. And what he found is, and that triggered this devotion to a whole issue on light pollution, that light pollution has actually increased by far faster than was anticipated. And he suggests that there's a global average increase in light pollution by almost 10%. And if you look at the variation in North America, it's higher versus in Europe, for instance. But this was a staggering result. And it goes back to, now we're starting with a bit of a historical review here, of course, to the invention of the incandescent light bulb, which was invented by Thomas Edison, one of the greatest inventors of all times, in 1880, and has led to a race to electrify the world, if you will, so that today we are here in a situation that because of all this artificial light, we can make the night seem like daylight. I always also like to point out, because I currently am in Vienna, that the very first implementation of the artificial light, or the light bulb that Edison invented, was taking place very close to Vienna in Brno, in the Stadttheater, the city's theater there. So what we have today, because of this invention, is that, the stars are disappearing as one effect of light pollution. Here you see how it would have looked like, or will look like, under excellent dark sky conditions, and on the right side, what you typically see nowadays in inner city skies. And one thing that was highlighted in the article that we were invited to write on the effects of this light pollution on human health is of importance, and it is a discovery that not only there are specific receptors in the retina responsible for transmitting this light exposure, but that also, particularly in the blue wavelength range, this light exposure will have more daunting effects on human health. So in 2017, the Nurses' Health Study set out, you see every dot here on this map is one nurse's address. And there's 120,000 nurses that are part of this large cohort study. And the goal was to see whether light pollution was associated with breast cancer risk in this nurses cohort. How this was done? Well, a lot of effort to geocode each of these addresses, and then using satellite data from the US defense system, basically down to 1 square kilometer scale, to be able to tell how much light exposure there was at each of these residential addresses and how it changed throughout time. And so the findings from this study was that there was a significantly increased risk of breast cancer among the women that in the top 20% of light exposure, so in the top quintile here. And this was significant and of importance, and was replicated and has been found before also by other studies. Here is another more recent example from Slovenia, where it was shown that particularly for breast cancer, they also looked at prostate cancer, where they did not find much of an association between light pollution, but for breast cancer, there was a higher rate of breast cancer among those who lived in areas highly polluted with light. But there were also, and are also, findings that are not so clear. There's a study from Denmark here that shows no clear association between light pollution and breast cancer risk here. And then there was this one particular study, which I found highly interesting, is based in the MCC, the Spanish case control study, where they examined light pollution and were able to distinguish between visible and blue enriched light spectra. And using this definition of light exposure, and we just heard, and you know that, of course, anyways, that particularly the blue enriched light will suppress melatonin at night, that in this category of blue enriched light, as a melatonin suppression index, in a way, there was a significantly increased risk of breast cancer. So a very detailed and intricate way to look at this. And today, to my knowledge, probably the best of these light pollution studies in terms of defining light pollution as it concerns the circadian system. Well, there's a totality of several studies now. This is probably needs to be added by the ones that I just showed you and a few more. But the basic message here is that outdoor light at night, which is what we call light pollution, shows by and large a significantly increased risk by 12% among the women with the higher levels of exposure to light pollution, this risk of breast cancer. Here is also indoor light exposure at night, which of course is also significantly elevated, but for light pollution, this here is our main concern. So that was a little bit of an opening just to show how we circled back again and again to this topic of light exposure, currently more in the form of light pollution. But how did all of this research get started? I will start off with the melatonin hypothesis. For those of you who don't know what the melatonin hypothesis is or was, can tell you, for me, it was quite relevant for my research. In fact, it goes back to what we now know, that blue light exposure falls onto the retinal receptor. And when the melatonin hypothesis was coined, really important to know that this whole system here was not known yet. So the melatonin hypothesis was actually indeed, sorry, I'm sort of getting kicked out here. Can you still see my slides?
Yes. Now you're back.
Yeah, okay, but I
No.
cannot navigate them. Maybe I should just stop share and share again. Maybe that helps. Let me try. So let me see. I'm not able to move forward with my slides, which I'm not exactly sure. Let me see why. It shows me the Safari can't find server, kind of as if I was online. So once more now. Well, let's hope. Okay, I hope it's working. Yes.
Yes.
So
Good.
coming back to this melatonin hypothesis, where, as I just said, this whole system was not known yet. We're still talking about the times when flashlight was used to shine light behind the knee. This was actually done at Harvard Medical School, and it was thought that this light behind the knee would modify our circadian system, because this retinal receptor was only discovered in 2003 by Bud Brainard and others. So this melatonin hypothesis goes back to the early 90s, late 80s of last century. And it was a hypothesis saying that if there's light exposure around 2 o'clock in the morning or in the middle of the night, as for example is the case during night work, that there would be a suppression of the nightly melatonin peak, aka circadian disruption, and that this lowering of melatonin would lead to a compensatory increase in estrogen levels, these circuits that we often see with hormones, that if you lower one, the other one gets increased. So the idea was, by lowering melatonin, estrogen levels will go up. And that was already known back then, that higher estrogen levels likely mean higher breast cancer risk. And this is the so-called melatonin hypothesis, which was in parallel also competed against by a Canadian group who were saying, well, it's actually not so much these compensatory increase of estrogen levels, but rather the lack of protection through melatonin that we think is the reason why night workers experience a risk of cancers, but not only breast cancer, but all cancers. So this is what was going on in the 1990s, early 1990s. And when I came to Harvard, this was in 1999, there were a handful of studies using registered data from Scandinavia, or case control studies, one by Scott Davis, where they measured light exposure indoors and related it to breast cancer, but there was no longitudinal cohort study yet that had examined whether light at night would be associated with breast cancer risk, to sort of bolster this melatonin hypothesis. And so I was at the Harvard Medical School, where these cohorts are located, 3 of them today. When I was there, nurses 2 had already begun, nurses 3 not yet. These are 3 independent large cohort studies of more of 100,000 registered nurses, each of them, where this is still ongoing here and still enrolling. So with this large data set, we were able to distinguish between women who had worked no night shifts or a lot of night shifts. And we were able to address for the first time longitudinally whether night work would increase breast cancer risk, taking into account all sorts of potential confounders, which may not mean much to you if not an epidemiologist, but these confounders are really important variables like job stress, for instance, where people thought that they have hampered previous results. So if you're not able to take them into account, your results will be biased. Well, this study here was able to take it into account. And what we found is that there was a significantly higher risk after many years of night work, 20 to 30 years of night work, and you would see a statistically significantly increased risk of breast cancer in both of the cohorts that we had at hand. So, of course, if you have this indication now based on a really good study base that potentially the melatonin hypothesis could be indeed correct, then you want to look at mechanisms. You want to understand what drives this increased risk. Is it indeed the estrogens, as was suggested by Richard Stevens and Scott Davis in the 90s? Or is perhaps this melatonin receptor at the cell membrane responsible for any alterations in breast cancer risk? Because by melatonin binding on the receptor, it was shown that this could block the binding of the estrogen receptor complex to this response element in the nucleus. And so there were these mechanisms to be explored. And what we then did as a next step in cohort studies, in large population studies, is to try to develop a biomarker for the circadian system that had to be practical, so it could be applied in 100,000 women or more. And so we looked whether if you measure melatonin in the first morning urine among non-night workers, but this is now looking at day working women only, so that their sleep is not disrupted, whether first of all there's variation over the years. And we could show that if you measure melatonin repeatedly over 3 years, for instance, in this study here, that it stays relatively stable over time, which is a prerequisite for a cancer biomarker. And what we also saw is that if you ship your urine for measuring melatonin to the lab, that this urine or the melatonin level you measure in this urine stays relatively stable if you keep the urine cold or frozen and ship them within 2 days or so. And so this ICC, the interclass correlation over 3 years here, is in the range of 0.72, which is comparable to, for instance, cholesterol levels. If you measure cholesterol every year once over 3 years, it remains fairly stable in individuals. And so this was reassuring and made us think we could set out and use this biomarker to explore whether melatonin, as one would expect, is associated with breast cancer risk if the melatonin hypothesis is correct. And based on the hypothesis, what we would have expected is that if women naturally produce less melatonin, sort of like as if they were working night shifts and get the melatonin suppressed by night work or light at night, that these women would have a higher breast cancer risk, and by extension, that those with higher melatonin secretion would have a lower risk of breast cancer. And that's indeed what we found. This is just showing data here from, again, the Nurses' Health study, but it was also examined in several other cohorts, for instance, the ORDET, that's an Italian case control study, to name one other study. However, even though this was great news that we could use and even confirm what we would expect based on the night work and breast cancer findings using this melatonin biomarker, what we were not able to show is that melatonin correlated with these sex steroids, as was predicted by the melatonin hypothesis. We would have expected if melatonin levels are lower, that these estrogen levels are higher, and vice versa. But we did not see this in at least this initial data that we had back then. You see, this is from 2004. And we even went out of the way and conducted a randomized controlled study with oral melatonin supplementation in breast cancer survivors to see whether if you increase your melatonin, this would lower people's estrogen levels as a protective factor ultimately to be able to use melatonin as a cancer preventive agent in, for instance, night workers or other populations. But here, this was conducted in a fairly older population. We could not show either that melatonin supplementation would decrease estrogen levels, or for that matter, any of these hormone levels here. They remained fairly stable after 4 months of 3 milligram melatonin oral at night. What we did show, however, at least to show that melatonin worked, I guess, is that sleep quality in these women increased quite well. Not only sleep quality, but also a measure of daytime functioning and the total Pittsburgh Sleep Quality Index score, each significantly increased. And so this is also interesting news because it ensures that women with breast cancer can use melatonin fairly safely, since we documented virtually no side effects. Now, these were some of the initial steps. Of course, mechanistically, then you're thinking, okay, so if the circulating levels are not impacted by melatonin, the circulating estrogen or other sex steroid levels not impacted, then maybe this mechanism sits at the tissue level. And so we looked at a melatonin receptor measured in the tissue of breast cancer patients. Here again, using data from this vast data repository from the Nurses' Health Study, where we had over 500 postmenopausal women's breast tissue and 800 matched controls, and where we were measuring the melatonin receptor status and found that whether or not they were positive for melatonin receptor, the light blue, didn't really matter. So the effect of melatonin was visible, but it didn't matter whether or not these women had expression of melatonin receptor. Disappointing, but one other attempt to see what the mechanisms were that were driving what we saw in the studies. So in the meantime, more and more studies had accumulated, population-based studies like the Nurses' Health Study, looking at night work and breast cancer risk. And so the IARC, for those of you who don't know IARC, it's a WHO subsidiary, the International Agency for Cancer Research in Lyon, in France. And they write monographs on potentially toxic or carcinogenic compounds. They also, for the first time, reviewed night work as potentially carcinogenic in 2007, and then again in 2019. There is a classification as to the carcinogenicity, and to cut the story short, night work was classified in both instances as a class 2A probable carcinogen, not class 1 definitive carcinogen, but probable. And I will tell you in a minute why it was classified as such. What happens at these IARC assessments is the whole literature that exists up to these time points is reviewed and then synthesized. And it's not only human evidence, but also mechanistic studies. The human evidence at that time point here, and these are all the experts that were at the second assessment in 2019, showed that night work, which it was defined there as involving work, including transmeridian air travel during the regular sleeping hours of the general population. And it was found that in these population studies, there was tremendous variability in how night work, the exposure was assessed, some with very detailed, some with less detailed assessments. And it turned out very atypical for epidemiology in many ways, that the case control studies, which we often refer to as the more potential for bias type studies if they are retrospective, that these had more detailed exposure information on night work than the old long-running cohort studies like the Nurses' Health Study that began in 1989. And the questions on night work were very crude because back then there wasn't really a hypothesis. It was almost golden coincidence, or I should rather say probably really smart foresight by Chuck Seisler and others to put this question on the Nurses' Health Study questionnaire. So anyways, what it was found at this 2019 IARC assessment was that a case control study that was lumping together multiple case control studies, so fairly big size. First author was Cordina Diverge, Emily, that had the highest quality and exposure information. And I just want to briefly zoom in onto that study because it was highlighted at the monograph meeting. What it had is not only duration of night work exposure, And you can see the top category was 20 plus years of night work. It also had information on the number of night hours per week worked and the intensity of night work, meaning how many night shifts or nights per week did the individual woman work. And the interesting findings here are among the younger women who are actually still in the workforce, as opposed to post-menopausal women who are getting into this retirement range. And we will see in a minute that this may actually impact the findings if you stop working night shifts. But here, these women were still actively engaged in night work, but had a long history for some part or high intensity. And by each of these parameters, it seemed like there's a significantly or almost significantly increased risk for breast cancer. And so this was considered as highly compelling in conjunction, of course, with these many other studies who were not entirely consistent, but still the majority was pointing towards an increased risk of breast cancer. So as I said earlier, again, the question on mechanisms, of course, will be considered here too. If you find an increased risk in an observational study for cancer, you want to confirm that the mechanisms that you would hypothesize to be underlying, that they indeed have been shown to exist. And vary according to the exposure, in this case, night work. So here's a bit of a summary of some of these studies, mostly to point out the scarcity of the data still, and that there is a need for more of these types of studies, which was also the conclusion of at the monograph meeting. So here, for instance, looking at the impact of night work on methylation, particularly methylation of circadian genes. There was a study by Jennifer Ritonia in the Bati lab conducted. Small studies, by and large, is what I will show you, because no really large studies haven't yet been published. But 74 female healthcare workers measuring methylation across 22 circadian genes and suggesting that there was night work was associated with differential methylation in some of these important circadian genes, including RORA, PER3, and RNTL. So there were also some other findings suggesting that night work impacts methylation. And this was followed by a study from last year where they conducted a genome-wide methylation analysis, again, fairly small size, 174 night workers. Of those, 58 non-shift workers, 58 short-term, and 58 intermediate-term night shift workers. And what they found is that there were some methylation positions that were differentially methylated according to shift work status, but the increased age acceleration that was linked to some of these pathways was observed only in the short-term night shift workers. And so it was thought that perhaps this is not a true finding, perhaps, but something, the so-called healthy worker effect, where only healthy workers remain in night work and those who sort of develop diseases drop out. Or perhaps more interesting, this finding could be an indication of adaptation to night shift work, something that is very interesting today, and we'll hear at the end of this presentation a little bit more about it. So then another potential mechanism that would link night work to cancer risk, and in particular also breast cancer risk, is oxidative stress. Here, for instance, is a summary of 12, all of them cross-sectional studies. And this is important. Cross-sectional means everything was collected at the same time, and that's usually not so ideal. You want longitudinal studies, but this is all that's there. And these 12 cross-sectional studies suggested that a night work was associated with several measures of oxidative stress. Importantly, increased DNA damage, but also DNA repair capacity and reactive oxygen species, to name a few. So this would suggest that there is a possible link. So I skipped back, I will skip forward now. And there's also one recent, even smaller, but interesting pilot study on night work and oxidative damage here, showing that levels of this measure of oxidative damage here, 8-oxo-DG, was significantly increased during the night shift period. And that this increase didn't go away immediately after stopping night work, but remained elevated after 1 month of discontinuing night work. Another indicator that would mechanistically support that indeed this link that we observe between night work and breast cancer risk is plausible is looking at immune cells counts, for instance, as a marker of the immune system. And here is a cross-sectional study again. You now know already that longitudinal would be more desirable. But in this cross-sectional study that is fairly large here, has 1,000 night workers and 10,000 non-night workers, all sorts of blood measures were measured and also absolute cell counts of leukocytes. And in that study, it was found that the night shift workers did have increased levels of some of these immune markers here, significantly increased levels, particularly leukocytes, but monocytes, and also, for instance, here, the basophil granulocytes. So some indication that night work impacts the immune system based on the measure of immune cell counts. And finally, the hormones. If we think about melatonin hypothesis, it was triggered by thinking that changes in hormone levels occur by suppressing melatonin. And so here in this study, this was conducted among men. So you have to think, okay, hormones, if they're relevant in male cancers, they probably are also relevant in female hormonally related cancers. So Harding examined here in a small study of 44 male factory workers whether night work changed these markers of male hormonal functioning, including adrenal androgen or progesterone. And there was again some indication in this very small study that night work might alter some of these hormones at least. So when you look, this is a nice summary of the various hormones that were examined in relation to night work. If the dot is on the left of this bar, it means that the level of this hormone, in this particular case, it's melatonin, AMT6 is sulfatoxin melatonin is lower. If the dot is on the right, it means the level is increased. So what we see here significantly increased was androsterone, and significantly decreased was progesterone and melatonin on the top here. So yeah, now there is a bit of a more indication that potentially melatonin suppression can increase maybe at least one hormone. These are the types of study that we will need more of them, and that we will ultimately have all these measures that I mentioned here in a systems biology approach where we can try to look at a variety of different pathways, including all the omics that we are now able to assess. So this is what the future should and will most likely hold. And what we take away from the IARC, the monograph assessment that was done by WHO on night work and breast cancer risk, is that yes, there is a lot of human studies, mostly indicative of a potential association between night work and breast cancer risk, but that there's not enough mechanistic evidence yet. Therefore, a class 1 classification was not possible at the time, and there is really a need for conducting these studies now, and also making sure that many of them are longitudinal studies, not only cross-sectional. So there's a recent study that sort of weaves us now into this other field of here subgroup phenotyping, with the idea that perhaps we can delineate the night worker with the highest risk based on whatever variables, mostly genetic makeup or the chronotype or degree of circadian disruption. This is a very interesting topic right now. And so to that end, a study that really had needed to be done for a long time and was recently completed was done in the Finnish twin cohort study, which some of you may actually know. It's a long-running cohort study that started in 1975. And it included mono and dizygotic twins that were selected from the Central Population Registry of Finland. The zygosity was determined by a validated questionnaire. So it's a really cool study design with questionnaires that were mailed to these twins. The surveys, the first one was mailed the first follow-up survey was made in 1981, and you can see that the response rate was really high, and it included detailed information on socio-demographic, psychosocial health, and lifestyle factors. And the night shift work definition in this Finnish twin study was self-reported from 1990, and looking at shift work patterns based on the current or latest work type. And so based on what we had, we defined rotating shift work in 2-shift or 3-shift patterns. So that we had in the end 3 categories. Women who worked fixed days only, so no night shift whatsoever. Then women who worked rotating shifts, but not including night work, so maybe evenings and morning shifts or whatever, but no night shift involved. And then rotating 2 or 3 shifts with night work, or fixed night shifts, so even more night shifts involved. So, and then breast cancer was assessed using the Danish registers, particularly the Finnish cancer registry, where all these cases are perfectly validated. And so what we ended up having is a sample of a couple thousand women, who most of them were day workers, but some of them also working night and 3 shifts. And they were of medium age, premenopausal still for the most part. And you can see here the characteristics distributed fairly evenly, with a relatively high smoking rate compared, for instance, to the Nurses' Health Study. And what were the main results of this finding, of this analysis? Well, most interesting, of course, was, for one, whether the night work breast cancer association was replicable also in the Finnish twin cohort, and it was. As you can see here, the 3 shifts or night only had a 58% significantly increased risk of breast cancer. And this was after adjusting for a variety of important breast cancer risk factors, as usually is done in these more detailed cohort studies. Interesting finding from the Finnish twin study was that there was a cohort effect. You can see here that among the women who were born between 1950 to 57, the risk of breast cancer was significantly higher according to their night work exposure than compared to the women who were born earlier in the 1930s and 1940s. Here you can see the difference really became larger in the more recent cohort of women. And because parity or oral contraceptive use, we were able to adjust for and sort of rule out a little bit that this was the main driving factor, that there were differences in Finland on these variables compared to other countries. But they didn't really change this. What might be the reason behind this is, if you remember what I said earlier, is that if women stop working night shifts, we have seen in other endpoint studies that the risk may tend to go down after a while. A little bit like if you quit smoking, your lung cancer risk eventually comes back to baseline. And so it could be that the women born in the 1930s had so long ago stopped working, because they are now long retired compared to these women here, that we start seeing this decline in risk after they had stopped working night shifts. So this is the explanation that we came up with. And of course, what we were also interested in is these sort of twin pairs discordant for history of night work and for breast cancer. And there were a couple of these discordant pairs. And what we found is that there was an increased risk here, 48%, not statistically significant risk in these matched pair data here. And when you conducted the analysis taking into account these discordant pairs, then the risk became slightly smaller, but was still there. And this suggests that the adjustment for other variables didn't change much, and that night work remains an important risk factor even after taking into account, as in this study, fairly well, for the genetic and family environment that may predict some of these risk factors for both breast cancer, but also perhaps for starting night work. And so where are we today? We are now heading towards individual risk assessment, precision medicine, precision prevention. These are tailored interventions. These are the keywords today, and they also apply to this research here, I would say. There is a little bit of evidence that I can show you that has already been produced, but I think for the most part, we are in the midst of trying to unravel here whether we can determine who the women are who are at the highest risk of developing breast cancer if they are exposed to circadian disruption. And I do want to remind us on this celebratory event in 2017, when Hall, Rosbach, and Young got the Nobel Prize awarded for discovering these molecular mechanisms, the genetics underlying the circadian system. It was really a great moment in time. And it was, of course, already long before studied, because the findings had been known to the scientists. There's more studies. I have picked a few to just highlight what had been known already 10 years earlier, before the monograph evaluation took place at IARC. Here, for instance, is a study in the Nurses' Health Study, again, looking at some pre-selected circadian genes and seeing how they were associated with breast cancer risk. And what was found is that if they had these genes, that sometimes, even after only 2 years of night work, their risk could go up almost threefold compared to women who didn't have this genetic risk. So these were some early indications. And this is one of the more recent studies that has been looking, for instance, at polymorphisms in genes that are relevant for melatonin biosynthesis and signaling. It's a fairly large study also among 44,000 women, and was conducted in the Breast Cancer Association Consortium, again picking predefined 97 candidates, single nucleotide polymorphisms, SNPs, at some defined regions. And what they found is that the TBH2 gene, tryptophan, is a precursor of melatonin, and the mitogen-activated protein kinase 8, the MAPK8 gene, were significantly associated with breast cancer risk in this study. And so these findings also do support a light at night hypothesis for breast cancer. Looking at some other important modifier, which also is very largely driven by genetics, is chronotype, or morningness, eveningness. And so here is one of the earliest findings from a field study where light measurements were conducted among women working night shifts or not working night shifts. So this here is women working 3 day shifts in a row with each, this is melatonin levels that peak at night, which each of these peaks representing 1 day. So the measures were going on for 3 days. And here you have 3 day measures of women who were working 3 night shifts in a row. And so what was really striking already in these very early findings is that you have here the chronotypes in different colors, that in green, the evening types here had, in the day workers, so regular hours, no circadian disruption, presumably, had quite distinctly different melatonin rhythms compared to the morning and intermediate types, blue the morning types and red the intermediate types. But if they are exposed to night work, this pattern reverses quite dramatically, suggesting that there is an interaction between night work and chronotype here. This has been studied, not a lot, especially for breast cancer. There's not very much evidence yet. But here is one study from Denmark by Johnny Hansen, who looked at the risk of breast cancer in night workers, stratifying by chronotype, where he shows that women with a morning chronotype, if they work night shifts and lots of cumulative night shift work, had a 4-fold increased risk of breast cancer compared to women with an evening type who worked a lot of cumulative night work. They had a 2-fold increased risk. So showing, these are some initial findings, even though they are way back, but there hasn't been so much new work done on this to kind of start delineating what drives the differences in risk that we see. There's something that you will hear more in 2 weeks, or when the last lecture is, by Angus Burns, probably, I'm thinking. But Andrew Phillips has done this work, really interesting, where he shows that there's a distinctive pattern in terms of sensitivity to light that can vary by factors of 50. And so here's, for instance, in this group of young individuals, when they start reacting to exposure to light at night can vary quite dramatically. Here's somebody with high sensitivity who reacts fairly quickly. Here's somebody with low sensitivity. I think this is highly interesting to build on these findings and be able to delineate what exactly will be driving as we move along discovering these things, the individual risks. And so I guess the question today is, when looking at night work and breast cancer risk, can we find, as actually not only for breast cancer, but for many endpoints that have been linked to circadian disruption, can we find a definition that allows us to improve our identification of individuals at risk? And these are some of the ways we have been trying to move towards this precision prevention. By looking at the genetic phenotype, initial work I showed you, there's work going on now to try to see whether shift work tolerance can be defined based on the genetic makeup. And hopefully these will produce interesting findings. There is what we have seen on chronotype and what a mismatch between chronotype and work hours can do to health, in particular, maybe cardiovascular disease, but maybe also important for breast cancer risk. And then whether or not baseline melatonin levels, because they can be quite different among individuals, are a sign of a healthy circadian system or of a more rigid or a more flexible system needs to be explored more deeply. Sleep regularity is being explored as a driving factor of the stability of the circadian system, perhaps. And then what other determinants do we have of this inter-individual variability in light sensitivity or sensitivity to circadian disruption? And so with this, I will now stop. I need to acknowledge, of course, the many fundings that I have received over the last years, most of it from the NIH in the United States, more recently also from the European Research Council here in Europe. And I do want to thank you for your attention and want to stop here because this group of people has done so much work while I was still in Boston. Still, to me, a really cool group. Each of them went on to do their great things. And so I will stop here and look forward to your questions.